This is part 2 of the pH lab notes. Here I've listed 3 liquids and their concentration of protons. For regular water, I have your 10 to the negative 7th molar. Another way to express this would be in its standard notation. But scientists don't want to write so many zeros. We don't want to write out so many zeros. So that's why we created this pH scale, and this helps us, simplify things. So what is pH? One definition for pH is the negative log base 10, that's important. And then here goes the concentration of your protons. So let's take water, for example. According to my data, the concentration of protons in H2O was this guy, 10 to the negative 7th. So let me plug that into my pH equation. Negative log base 10, 10 to the negative 7. And that number just came from right there. What this part of the equation tells me to do, it's basically asking me 10 to what power, so 10 to the question mark, is going to give me what's on this inside, 10 to the negative 7. So common base tells me the exponents are the same. So my question mark here is equal to negative 7. So pH, let's not forget my negative sign from up here. And then this is equal to that. So pH of water is 7. So again, we use a pH scale. We use pH to help us simplify things, easier to present. So let me write out my scale. We have 0, in the middle we have 7, and at the end is 14. You could have below 0 and above 14, but for now we'll just deal with this. Around 7, we call this neutral pH. If you ever dip below 7, this is acidic. And if you're ever above 7, we call this basic. Excuse me. Basic. And what does that mean? So if you dip below 7, so if we go towards the left, we have more protons. Or you can think of it as less hydroxides. Now if you go towards the right, you have less protons and you have more hydroxides. Okay, so 2 things to keep in mind when dealing with pH. If you ever take the concentration of protons, and then if you multiply that by the concentration of hydroxide, this will give you 10 to the negative 14. Now, if you take log on both sides, the negative log, take the negative log on both sides. I won't show the math here, but this is basically going to give you pH plus POH gives you 14. And now you can use these 2 equations to help you with pH problems. Okay, so on to buffers. A buffer consists, there's 2 ways to make one. One is with a weak acid, so something that doesn't dissociate completely, and a strong base. Or vice versa, you can use a weak base and a strong acid. In today's lab, you worked with a combination of this. So in this new solution you created, there is a pH range where it's going to resist changes to pH. So if you keep adding acid or if you keep adding base, the pH isn't going to change that much. We call this range the buffer zone. And then kind of in the middle of the buffer zone, there's a certain pH where the buffering is greatest. We call this the pKa. And remember, the pKa is just a pH. It's a certain pH, unique to acids and bases. And to help you find the buffer zone, it's basically plus minus 1 of the pKa. So what do I mean by buffers, how they work? So if you were to add a strong base, so if I were to add OHs to my solution, normally without a buffer, this is going to increase the pH significantly. Increase the pH significantly. There's nothing holding it back. But if I were to add hydroxide to a buffer, and let me give an example. So CH3COOH, this is acetic acid. This dissociates a little bit, gives up a proton. This is what I have. So now if I were to add OH to this proton here is going to take care of this OH, neutralizing it. And this is going to give us H2O. But I want you to note here that the OH was neutralized. OH neutralized. So the pH will not change as much. So as long as these protons are here, it's going to sop up these OHs until it's gone. So for this case, we'll sop up hydroxides, keeping pH from changing. We'll sop up hydroxides, keep the pH from changing until the protons are gone, until all protons are used up. Until all protons are used up. And then we'll see significant changes in the pH. Significant changes in pH. And the process of adding a strong acid or a strong base to a solution, this is called titration. And that's what we did in today's lab.